Understanding the mechanism of the catalytic reaction and the nature of the key active sites on the surface of the catalyst is a prerequisite to the truly rational design of the efficient high-performance catalytic materials. An advantage of the electrocatalytic mode of catalysis over other types of reactions is that we can actually probe the key charge transfer events occurring at the surface of our materials that support and drive the overall reaction. However, no matter how sensitive the electroanalytical tools we use are, they only provide an indirect information on the chemical nature of the active sites of the catalyst. So we cannot really understand what is the chemical nature of the charge transfer reactions we can measure. The only direct way to probe this is to employ the in situ and operando methods of analysis. In other words, probe the active species spectroscopically under the electrochemical conditions for the catalysts in action. In this way, we can identify the active sites of our catalyst and understand how we can make it better. Moreover, operando analysis allows for the detection of the weak sides of the catalyst, so we can study the most of the degradation of materials, which is also a very important piece of knowledge in terms of the development of efficient catalysts. Therefore, in situ and operando methods provide a truly useful tool for the investigation of the electrocatalytic materials and is indispensable for the design of the efficient new catalysts. Over the last years, ACS researchers at Monash have been actively developing different modes of the in situ spectral electroanalytical analysis to support our studies of the catalysts in action and to understand the modes of their operation and degradation. Traditionally, we've been focusing on the X-ray absorption spectroscopy, which uses photons with the tunable energy to probe the structure and the electronic properties of our catalysts. Most recently, we decided to extend the arsenal of methods available to us to the in situ X-ray photoelectron spectroscopy. Unfortunately, these kind of experiments are not yet possible at the Australian synchrotron, so we had to travel to Berlin, to the BESSY Synchrotron Facility, where the leading researchers in the field have already installed and thoroughly tested an in situ electrochemical X-ray photoelectron spectroscopy setup at their high-end end station at the ICES beamline. We have secured a beam time, and in October, ACES PhD candidate Rebecca Hodges and myself have traveled there to undertake the experiments. Additionally, we were actively supported by the past ACES graduate, Dr. Shannon Bonke, who is currently the Alexander von Humboldt Fellow and the Max Planck Institute for the Chemical Energy Conversion in Mulheim. Classical layout of the X-ray spectral electrochemical cell uses the working electrode both as the support, the substrate for our electrocatalyst of interest, and also as the barrier between the X-ray chamber, which is often required to be under the high vacuum conditions, and the electrochemical chamber, where our process of interest occurs. So what happens during the analysis? We apply to the potential to our working electrode, X-rays come in, then interact with our catalyst, and then other X-rays are emitted, which we analyze and get structural information and also information on the electronic properties of the material. However, as follows from the name of the XPS, in this case we need to detect photoelectrons. And those cannot really go through the commonly employed electrode materials for the in situ work. So these are typically 50 to 150 nanometer thick silicon nitride or the silicon carbide membranes. Electrons cannot go through. So the solution to this problem was introduced several years ago by using the Holy membranes, which have the holes there. And in this case, as the working electrode, we are using two layers of graphene, which serve both as the working electrode and as a separator between the X-ray chamber and the electrochemical environment. So in this case, X-rays come in, interact with the catalyst, and those emit electrons, which we can now detect and analyze in a meaningful manner. Thus, we were able to record X-ray photoelectron spectral of our catalyst as a function of the potential applied. Moreover, the use of the synchrotron radiation with the tunable energy allows for significantly enhanced sensitivity of XPS towards specific elements like, for example, nitrogen, which was of special interest to our experiments, which were focused on the nitrogen reduction reaction and absorption of nitrogen species on different catalytic surfaces. Moreover, in this configuration, we can still do X-ray absorption spectroscopy analysis, which provides the complementary and very useful set of information taken together with the in situ XPS. After 1 week of intense work, ACES researchers have managed to generate a significant array of new data. And equally importantly, we have gained unique training and new skills and knowledge in the field of the in situ X-ray spectroscopic analysis of the electromaterials in action. Furthermore, we have strengthened our links to the world-leading researchers in the field. The specialists at the BESSY Synchrotron Facility provide a unique environment for the analysis of the electromaterials, either ex situ or in situ. Our ongoing aim is to ensure that the Australian researchers have access to the best world-class frontier techniques and facilities for the analysis and characterization of the electromaterials.